and bond reduces, and all of a sudden you do start to see this breakdown of the gel, and here you can see it's starting to get a lot more opaque. So if you want to quantify that, we have done so. The gel degradation on the left, this is the weight percent, and then this is time. And if it's just in a PBS buffer, it stays very constant, shown here in green. But if you put it into this glutathione or reducing agent, you can see it loses a lot of weight over time. And then you can think about using this for drug delivery. So on the right, we have we encapsulated dextrin kind of as a model biomacromolecule. You can see the cumulative release of dextrin on the y-axis over time. In PBS, you do get some loss, but you see kind of this acceleration of the loss if you put it into that reducing environment. The next slide is kind of a fudge because I think it's a really cool behavior, but we actually did this with polyethylene glycol instead of recombinant proteins. So you see we have a PEG with thiols, and we use this DVS or this FeEDTA. And now the idea here is that both of these cross-linkers are actually working with the thiols, but sometimes you're going to get this disulfide bond, right? So that's reducible and reversible, or you'll have a thiol ether bond, which is a covalent bond, which you can't actually reverse. So the idea here is that by using these two different cross-linkers, you're basically cross-linking the same thiols from your PEG, but you can vary the ratio of how many of those bonds are actually reducible or reversible. So the reason we thought this would be interesting is because cells really do respond to mechanical properties, and we can basically decouple a little bit how easily you can degrade the material versus what its initial stiffness is. So if you look here at the elastic modulus on the y-axis, and then we're keeping the FeEDTA3 concentration constant, but we're varying the DVS concentration. So it goes from 0 to 8. And you can see we're not varying too much. We're not, the range of elastic moduli is between 10 and 22 or so. So our moduli has not changed too much, but our degradation and the way it could deliver drugs has changed. So here, this is our cumulative release. And this is over time, and the solid line is with PBS, and then you have the dotted line with PBS with the reducing agent. And so if you have 8 millimeters of the DVS, you can see there's not very much difference. But then as you lower the amount of DVS and you have more reducible bonds, you can see that at 6 millimolars, you have a lot more degradation when you add that glutathione, so there's a bigger difference. And then when you have 4, you again have this big difference. And then 2, you can see there's a big difference. So you can basically start thinking about, do you want to have a similar initial stiffness, but be able to tune the degradation? That could be used for drug delivery, or it could also be used for cell studies, particularly because cells do respond to the mechanical environment a lot. And then kind of the last thing I wanted to show you with our materials development was some other tools we've used for tissue engineering. We have put in this bone morphogenetic protein 2, the BMP2 peptide, and when it's incorporated within the backbone of our proteins and the authentic sequence, and we put stem cells on there, we do see this really bright pink, alizarin red staining. Whereas if we put the peptide with the same amino acids, but we kind of scramble up the order, you can see that the cells can tell a difference because they're not really undergoing bone differentiation as rapidly. We also looked at vascular, so endothelial cells. And if you look at the mechanics, if you have a soft gel, the cells are not very spread as you would expect, and the stiffer gels are a little bit more spread out, but that can also correlate to endothelial function. So here we were staining for a function characteristic of endothelial cells. They intake this AC LDL molecule. So on the soft gels, they don't take in very much, whereas here you can see there's a brighter fluorescent staining because they really are taking in more when they're on a stiffer gel. And then when we incorporate that vascular peptide, so this QK is the peptide that is derived from part of VEGF, you can see that we do get the cells starting to form these like extensions to make vessels. But you can also look at kind of this vascular function again, and these are proliferating cells, so it's low staining, negative control, very low staining, but you're starting to see a lot more of that function, that internalization of the AC-LDL when the cells are on proteins that have that peptide in the backbone. And then this is a positive control where we add VEGF. So this is my second story, and I hope that you've seen that the recombinant proteins really allow you to design materials tailored for specific applications, and you can make materials that respond to stimuli such as pH, temperature, reducing environments, and then you can also kind of add in these peptides to influence cell differentiation. So for my last story, it's a collaboration that we have with Jonathan Wilker over in Purdue Chemistry, and this is based a lot on muscle adhesive chemistry. So you can see lots of these muscles. They adhere to different surfaces by secreting these threads, and then at the end of the threads are these plaques. And the plaques are full of these muscle adhesive proteins or MAPs that have this DOPA molecule that's been found to really contribute to adhesion underwater into different surfaces. So when you think about a motivation, right, we have over 200 million major surgeries a year, and they require wound closure with sutures or staples. I don't know if you've had either sutures or staples recently, but they do hurt. I remember I had them a few years ago, and it does hurt. And you're poking holes into the tissue, you're opening yourself up to infection or scarring in particular. So having a surgical glue would be easier to apply. Now, when you're thinking about making a surgical adhesive or glue, you do want it to be biocompatible. You want it to work in a wet environment, particularly your body has a lot of fluids or blood. You do want strong bonds, so that means you want it to adhere well to the surfaces, but cohere or stay intact together. And then for thinking about cell responses, you do often want the stiffness or the rigidity to match the surrounding tissue. Now, one of the challenges that we particularly thought about is if you do have these underwater adhesives, in general, anytime you add water, a glue is not going to work as well. And another challenge of underwater adhesion is that if you're applying the glue, you want it to stay in place. You don't want it to kind of disperse into the surrounding fluid. And so there had been a couple of studies before. This is from our collaborators lab. This is an underwater picture where they're dispensing the glue in here, but it's in chloroform, and that's how it's partly staying in place because of the density difference. Another example of a delivery system is you can use a coacervate adhesive, and this was inspired by the sandcastle worm. So the idea here is they had two different polyelectrolytes, and at pH 4, it's a well-dispersed solution, but at pH 7.4, the oppositely charged molecules will come together. It'll form this very dense liquid phase where it's polymer rich at the bottom, and then kind of a polymer poor phase, and they tipped it onto its side to show you that it is a liquid and not a solid phase. So in terms of what we decided to do is we kind of went to that recombinant protein idea, and we said, okay, well, we can take the idea of having the DOPA molecules from muscle adhesive proteins that confer basically adhesion in a wet environment. They're biocompatible, they have strong bonds, and we've worked a lot with these elastin proteins. Now, elastin proteins, I kind of alluded to earlier, they're elastomeric or flexible, but they're biocompatible, and they have this cool property that by themselves, not with a partner, but by themselves, they undergo this reversible coacervation. So at cold temperatures, they're very soluble, so it's kind of showing you a clear solution here. And if you warm it up, and you can kind of tune this temperature to body temperature, say, if you warm them up, you do have this phase-phase separation where you get this dense liquid phase and kind of a polymer poor phase, right? And so that dense phase would allow the glue to stay in place in the solution. Okay, so to actually kind of visualize what we did was we combined this smart coacervation from elastin with DOPA's wet adhesion, and we actually just designed a gene that had elastin sequences interspersed with these tyrosines. We also put amines in as potential cross-linking sites. And then we used an enzyme to convert the tyrosines into that DOPA molecule. And then now we have two chemical moieties we can kind of play with. We have the amines that we can do cross-linking with, and the DOPAs can also undergo oxidation or chelation. And then that's kind of our glue. So what we did was, first we wanted to see, are cells viable, right? If we're thinking about biomedical applications. So we looked at viability. We have cells on a positive control. YKV is our unmodified protein, so the ones that have the tyrosine residues. And the MYKV is the modified version, so that has DOPA residues. And we're seeing really high viability in both. We were also, since I mentioned to you, we wanted to match stiffness of soft tissue. We looked at our Young's modulus and we did different cross-linking ratios using that amine. And we see that we can tune basically the modulus within the range of soft tissue, which is about 2 to 200 kilopascals. Okay, and then the next thing we really wanted to see is, well, what kind of adhesion do we get? The first test we did is kind of this lap shear adhesion test. You take two substrates, you put the glue where my palms are, you let it cure, and then you pull. And you're trying to measure that maximum force that it takes to pull it apart. We did start in this first study with aluminum just because it's a little bit, it's more standard and the variability is lower. And what we found is when we cure in a kind of a dry environment where we don't add moisture, we have adhesion strength. BSA, bovine serum albumin, is a protein control. We don't expect it to stick, and it doesn't. Seal is an FDA-approved sealant. This is our unmodified protein, and this is our modified protein. So we're definitely seeing an enhancement with our proteins. But then we added water, and I went the wrong way. Sorry. We added water. So we had a humid environment for our cure. And here you can see, I will admit, right, look at this. If you look at this y-axis, this is an order of magnitude different compared to the dry cure. Anytime you add water, you're going to get less adhesion. All of a sudden, now you see why that DOPA matters, because the unmodified protein is as low as the Seal and the BSA, but the modified protein is giving you a stronger adhesion. And then I mentioned one of the reasons we picked elastins because of that coacervate property. I'm trying to show that here. Maybe you can see it. But at low temperatures, it's very clear. It starts to get cloudy as you heat it. And if you wait, it basically phase separates, and that polymer-rich portion is almost sticky like honey. And you can tune the temperature at which that transition happens. So this is what's being shown in the bottom. So normalized transmission, so you have transparent solutions where everything's soluble, and then you can increase the temperature. And then what you can see is if we're starting at 150 mg per mil and a pH of 7.5, shown here in this kind of blue color, if you take the pH away from the isoelectric point, you can increase the temperature that transition happens at. If you add salt, you can decrease the transition temperature. If you increase concentration, you decrease the temperature. And here, this kind of jagged line, this red one, is when you modify the tyrosine so it has DOPAs. And you're not getting as clean of a trace because when you do that enzymatic conversion, not every tyrosine is converted to DOPA. So you kind of have a collection of differentially modified proteins. But anyway, we did tune it to body temperature. And then this is the proof of concept. So here, this is a picture that you can kind of see. This is the air above, this is the liquid interface, and we're pipetting our protein glue down there. And if you look at the video, you can see this is completely underwater and the glue's staying in place. It's a water-soluble glue, but it's not just dispersing into the surrounding liquid because of that coacervation property. And that coacervation property allows us to actually use the glue underwater. You can see we were able to measure an adhesion strength of about 3 kilopascals. But if you have the unmodified protein, it doesn't stick very well. We couldn't, I'm reporting at 0, but basically we can't get a test because the adherents break apart before we get them to testing. To seal, the same thing. And BSA has nothing reported because it's water soluble, but there's no glue, right? It's dispersed into the surrounding liquid. So the last part of this story is something where we kind of wanted to dig a little deeper. So a lot of people have used the DOPA. based on the Mussel underwater adhesion, and you can see that reduced DOPA form can do adhesive surface binding to different substrates. It can be oxidized, and then it can start to make covalent bonds between itself. It can have this iron crosslinker here, and you can get different kinds of bonds, so cohesive binding in the bulk of the glue. But also, if you look at Mussel adhesion chemistry, they do have these proteins with thiol groups, and that can work as a partner protein to reduce the oxidized DOPA to the reduced form, or the proteins with the thiol groups can also interact with the oxidized DOPA to kind of form these adducts, so that you have this thiol connected to that DOPA group. And so we wanted to investigate, the different contributions of the thiol versus the DOPA chemistry, and how would that contribute to bonding. So just kind of a comparison, DOPA versus thiol. So the DOPA is over here. It's versatile in its chemistry, right? It can do chelation, hydrogen bonding, covalent bonding, but it is a bit tricky to incorporate, right? You have to put that tyrosine, then use an enzyme to convert it. Now, on the other hand, this thiol is easy to incorporate because you can just encode a cysteine amino acid. But there are some limited adhesion mechanisms, and mainly it's getting oxidized. If you really wanted to form a covalent bond, you might need some unnatural functional groups to add in. So this study really wanted to compare DOPA-based versus thiol-based adhesion. And we also wanted to then say, if we have both methods of adhesion, are we able to get higher or stronger bonds, essentially? So the first thing we did is, this is actually with pig skin tissue. So we moved away from the aluminum. We wanted to make it more relevant. And so because of that, and pig skin varies from batch to batch. So rather than reporting the actual adhesion strength, we're normalizing it to a certain glue because the pig skin will vary. And so here, this MYKV72, this is the ones that have just the DOPA. And then this long version, the key thing is that it starts with a C. So there's a cysteine residue, or it has a thiol. So when you just have the DOPA shown here in blue, you can see at 1 hour of curing, we don't get a lot of, we can't measure that adhesion, but we can measure adhesion at 6 and 24. But when we just have the thiol and no DOPA, we're getting actually really fast and stable adhesion with just 1 hour of cure time. And then the next thing I said we wanted to do is say, okay, thiols cure faster. Actually, in this particular instance, we're also getting higher adhesion strengths, but what if we combine the thiols with the DOPA chemistry? And then that's what's shown here in purple. So we have a protein that has the DOPA and also one that has the thiol. We added a crosslinker as well. And then I'm going to show you two different controls on this slide and the next. So this one in pink, instead of having DOPA, we've substituted tyrosine, but we do have the thiol proteins. So here we're really trying to see, compared to our control that has tyrosines, how much more does the DOPA add in terms of adhesion strength? And we varied kind of the DOPA to thiol ratio. And you can see at a 1 to 0.5, 1 to 2, we don't really see a difference, but at a 1 to 1, we see that the DOPA actually does increase adhesion. Now, if we take that same mixture and compare it to a control that does have the DOPA but does not have the thiols, we can now see how much more adhesion do we get if we have a thiol. So we're comparing the thiol DOPA mixture in purple to basically a glue that has DOPA but no thiols in this light blue. And in all of those molar ratios, you can actually see that having that thiol makes a big difference. We're getting a big statistical difference, right? So here we get this increase, particularly at the 1 to 1. And so it does seem as if the thiol contributes more to the adhesion strength in these DOPA-thiol mixtures than DOPA does itself. And then the last thing we wanted to do is say, look at these mixtures and say, well, how does it perform in terms of over time, right? Can we have shorter adhesion or shorter times and still get good adhesion strength? So in this purple, these are our mixtures, but they do not have the crosslinker. And then this dark purple, we do have this FE-EDTA crosslinker. So you can see we are getting really good adhesion strengths at the 1 hour time point. And having that cross-linker kind of decreased our dependence on time for our adhesion strengths. Okay, so that was my last slide about that story. And I hope that you've seen that combining both that adhesive domain with an elastin-based polypeptide produced a promising soft tissue adhesive. The thiol-based ELPs had faster curing and stronger adhesion compared to DOPA-based ELPs. The strongest formulas cured quickly and were stronger than to seal. But then really combining that DOPA and thiol cross-linking chemistry did increase adhesion strength. Obviously, I'd like to thank my group. They're wonderful. I've had some wonderful graduate students and postdocs. And then my collaborators, Alyssa Panich and Gert Brewer and Abby Cox, particularly on the cartilage work, and then Jonathan Wilker for the adhesive work. And then these are our funding sources. And now I'm happy to take any questions.
Thank you for this presentation, Julie. Appreciate it. Many questions came in. How do you explain the alkaline phosphate activity decrease with collagen 1 and 2? How does native collagen 2 help to prevent osteoblast formation?
Yeah, I mean, I don't know that people really understand the mechanism of why collagen 2 seems to be more chondrogenic, but it does seem to have that property. We've seen it in ours, right? So we see better cartilage matrix formation and then that lower osteogenic differentiation. And we had gotten this idea because other studies had seen it as well. I'm guessing that the cells are interacting with particular receptors with the collagen 2 and then activating kind of probably the SMAD pathways for cartilage differentiation, but we haven't dug into the actual mechanism.
Thank you. Can you please clarify why stiffer modulus leads to increased endothelial function in the DVS examples?
Yeah, I will also say that when I say stiffer, it's a relative term. So they're not really what you might consider stiff hydrogels. If I recall correctly, they're still in the kilopascal range. So it's just that they have enough, they're stiff enough so that the cells can actually spread. So endothelial cells themselves typically have that cobblestone morphology. So I would think that that would, having that morphology, again, we didn't investigate the particular mechanism, but I think that that will be coupled to having the endothelial function, right? Versus having this really rounded shape that I don't think is indicative or characteristic of endothelial cells.
Thank you. Wonderful presentation. Is it possible to oxidize DOPA via electrical potential? And can you reverse the oxidation using the same approach?
That's a really good question. We have not tried to do that, but I believe that I saw a paper in the literature that has started to investigate that. I think it could be interesting, but one thing that can be tricky is especially the oxidized DOPA tends to react very quickly. So if you have that form, it's going to start forming covalent bonds. So often one of the tricky parts of using these type of glues is actually keeping it in its reduced state before you want to use it, right? So people often use pH, right? They'll use low pH or maybe ascorbic acid to kind of keep in the reduced form so that it doesn't over oxidize, meaning oxidize, and then form those covalent bonds, after which you can't really reverse it.
Thank you. How does the pore size changing with DVS? Maybe a reason behind drug release kinetics.
Yes, I'm trying to remember. I don't think we looked in depth, but that is a good point. However, I think we do see the degradation. So I think the pore size would probably be more likely to influence the initial burst release. Certainly, it could have some effect as you continue, but I think that burst release would make a big difference. I think we also looked at some molecules that were smaller than and also on the order of the pore size or the estimated mesh size, I guess. We didn't measure the it depends. Like we didn't do a cryo and do pore size, but we estimated the mesh size based on the swelling that we're getting from those hydrogels. And I think, yeah, I'll leave it there. So I think it was really the burst release where the pore mesh size made a difference.
Thank you. In slide 21, how stable is the degradable gel in cell medium?
I have to see what slide 21 is. Sorry. Oh, this one. I would expect to have a very similar amount. It depends if you mean cell medium with or without FBS, right?
Yes.
We didn't do it in cell media, but I would imagine that the results would be very similar in media.
Thank you. Thank you for the great talk. In slide 36, why does the adhesion decline over time when thiol was used?
Oh, I think I know what you're talking about, but let me get there. Oh, are you talking about this, how it goes down? So technically, when they say AAA, they're actually statistically the same. So we're not seeing a decrease. I know the bars look like they're decreasing, but you'll see that the error is so large that this is actually the same value.
I see. Okay. Well, thank you for the fantastic talk. We got lots of questions. Appreciate your time, Julie.
Thank you.
Have a nice day.
You too.